That tiny letter on your medical chart may carry a story thousands of years old, shaped by migration, disease, chance, and survival. Blood group patterns can offer limited clues about population history and biological variation. When scientists compare blood group patterns across populations, one striking feature appears in parts of South. Regional and donor studies in the Philippines and Vietnam have reported type O frequencies above 40% in some samples. These figures, however, should not be treated as fixed national identities. Frequencies can vary by region, ethnicity, sample size, and whether researchers study blood donors or the wider population. The deeper value of these numbers lies not in ranking one country against another, but in understanding how inherited variation moves through time. The ABO gene contains versions passed from generation to generation, and scientists can compare them with other genetic markers to study migration, population mixing, genetic drift, and natural selection. Type O is not a genetic passport leading directly back to one continent. The ABO system is extremely ancient, with evolutionary roots that predate modern humans, and its major variants have persisted across many populations. Asia therefore offers not a single source code for humanity, but a rich genetic archive. Combined with whole genome research, archaeology, and ancient DNA, blood group data can contribute one useful clue to the much larger story of how people moved, mixed, adapted, and survived. Far to the north, a chapter of human migration unfolded around Beringia, the region that once connected northeastern Asia and Alaska during the last ice age. Archaeology, ancient DNA, and mitochondrial research indicate that the ancestors of indigenous peoples in the Americas descended largely from populations connected to Northeast Asia. Their journey was probably not a single march across a frozen bridge. Researchers continue to examine periods of isolation, multiple movements, and possible coastal routes as communities spread across the continents. Type O is remarkably common among many indigenous American populations. In some isolated communities, type O reached very high frequencies in some historically isolated populations. Even so, that pattern does not prove that every founding traveler had type O, nor can one blood group explain the settlement history of the Americas. A founder effect may have contributed. When a small population establishes a new community, the variants it carries can become disproportionately common in later generations. Population bottlenecks, genetic drift, and possibly disease-related selection may have strengthened the pattern. Type O is therefore a useful clue, but not a complete migration map. Only when blood group data are combined with ancient genomes, archaeology, and language can researchers build a picture of how founding populations expanded, adapted, and created extraordinary diversity. Across the Pacific, human expansion took a different form, not a journey over land, but a sequence of voyages between islands. One influential model suggests that Austronesian-speaking communities moved from Taiwan through the Philippines and island Southeast Asia before reaching remote Oceania. As they traveled, they mixed with populations already living across the region, creating the complex ancestry found among Pacific peoples today. Their descendants crossed enormous distances in canoes, using stars, winds, ocean swells, birds, and cloud patterns as navigational guides. Over generations, they settled places as distant as Hawaii, Aotearoa, New Zealand, and Rapa Nui, completing one of the most impressive maritime expansions in human history. ABO frequencies vary across Pacific populations. Type O is common in several communities, but it is not uniformly dominant from island to island. Founder effects, genetic drift, isolation, natural selection, and later population mixing can all shape these patterns. Similar blood group frequencies may also appear in distant populations without proving recent shared ancestry. Comparisons between Pacific Islanders and indigenous Americans therefore require stronger evidence than blood type alone. Ancient DNA, archaeology, linguistics, and whole genome studies provide a clearer picture. Some research suggests limited prehistoric contact between Polynesians and South Americans, but type O cannot reveal when, where, or how that contact occurred. At the dawn of the 20th century, blood transfusion was still frighteningly unpredictable. One patient might recover, while another could suffer a rapid and sometimes fatal reaction, and physicians had no reliable way to explain the difference. Karl Landsteiner changed that. Working in Vienna, the Austrian physician separated blood cells from serum and systematically mixed samples from different people. He noticed a clear pattern. Some combinations remained smooth, while others caused red blood cells to clump together, a reaction called agglutination. From these observations, he described 3 blood group patterns that were later standardized as A, B, and O. The fourth major group, AB, was identified soon afterward by Alfred von de Castello and Adriano Sterli. Together, these discoveries formed the ABO blood group system and gave medicine a practical way to understand why certain transfusions were compatible while others were dangerous. This was far more than a new label for hospital charts. It transformed transfusion from a gamble into a procedure that could be planned with far greater safety. Landsteiner's work later earned him the 1930 Nobel Prize in Physiology or Medicine. Today, the letters A, B, AB, and O may look ordinary, but they represent one of the breakthroughs that helped make modern surgery, emergency care, and transfusion medicine possible. The next surprise came from genetics. Science did not reveal type O as humanity's single original blood group. Instead, it showed that the history of ABO was far older and more complex than any simple ranking of A, B, and O. The ABO gene sits on chromosome 9 and provides instructions for enzymes that modify a structure called the H antigen. The A version adds one type of sugar, while the B version adds another. Many common O variants contain changes that disable the enzyme, leaving the H antigen largely unmodified. That may make O appear more primitive, but evolution does not move in a straight ladder from simple to advanced. Genetic comparisons among humans and other primates suggest that A-like and B-like variants are extraordinarily ancient, with some lineages persisting across species for millions of years. O variants, meanwhile, appear to have arisen more than once through separate mutations. Global blood type patterns were shaped by migration, population bottlenecks, genetic drift, and possibly selection from infectious disease. The same blood group can therefore become common in distant populations for different reasons. The genome replaced an appealing origin myth with a richer explanation. ABO diversity is not a hierarchy, but an ancient record of inheritance, chance, environment, population history, and survival. In 1952, a blood test in Bombay revealed a puzzle the standard ABO system could not explain. The patient's red cells appeared to be type O because they carried neither A nor B antigens. Yet further testing showed that type O blood was incompatible, creating a transfusion risk. The explanation lies beneath ABO labels. Think of the H antigen as the foundation of a house. The A and B genes produce enzymes that add finishing pieces to it. In most people with type O blood, those additions are absent, but the H foundation remains present. People with the Bombay phenotype are different. Because inherited changes affect the FUT1 gene, their cells do not form the H antigen. Without that foundation, A and B antigens cannot be built. Even if the person carries A or B versions of the ABO gene, basic forward typing may therefore make the blood resemble type O. The immune system can also produce anti-H antibodies, which may react strongly against ordinary type O red cells. For that reason, people with the Bombay phenotype require red cells from another compatible donor with the same phenotype. What appears to be an ordinary O result is therefore a rare and clinically important condition, showing why laboratory testing matters. One of the clearest examples of how type O may influence disease severity involves Plasmodium falciparum malaria. The finding does not show immunity. Instead, it reveals how differences on red blood cells may affect what happens after infection begins. The parasite can invade red blood cells from every ABO group. Once inside, infected cells may attach to healthy cells and form clusters called rosettes. These clusters can obstruct tiny blood vessels and are associated with severe complications because type O cells lack A and B antigens. Type O red cells have been associated with reduced rosetting in several studies. Population studies have therefore linked type O with a lower average risk of severe falciparum malaria, not with complete protection from infection, complications, or death. Norovirus tells a different story. These viruses interact with histoblood group antigens displayed on cells lining the digestive tract. Susceptibility depends heavily on the viral strain and on secretor status, which is influenced by the FUT2 gene. Type O is not a universally locked door. In some studies, secretor-positive people with type O were susceptible to several norovirus variants. Evolution often produces context-dependent advantages rather than universal protection. A trait may reduce one risk while offering no benefit against another. Blood type can shift probability, but it cannot predict individual outcomes. Protection associated with one disease can disappear in another, and type O provides an example. The same blood group pattern linked to a lower risk of severe falciparum malaria may be associated with greater vulnerability elsewhere. Cholera is one of the strongest examples. People with type O do not necessarily become infected more often with Vibrio cholerae, but studies have found that, once infected, population studies have associated type O with a higher average risk of severe cholera outcomes after infection. Researchers are still investigating the mechanism, although differences in how intestinal cells respond to cholera toxin may contribute. The connection with Helicobacter pylori is less straightforward. Type O has long been associated with a somewhat higher risk of peptic ulcer disease, but studies do not consistently show that people with type O are more likely to acquire the bacterium, bacterial strain, secretor status, medication use, smoking, and other health conditions also matter. Mosquito attraction is even more complicated. A few laboratory studies have reported possible ABO-related landing preferences, but the evidence remains inconsistent and cannot reliably predict who will be bitten most often. Body odor, carbon dioxide, heat, skin chemistry, movement, and mosquito species may be more important. These associations shift probabilities at the population level, while individual outcomes still depend on many biological and environmental factors. A blood type letter can influence clotting, but not because type O blood is simply thinner. The key difference involves proteins that help platelets attach and stabilize a clot. On average, people with type O tend to have lower von Willebrand factor and factor VIII levels than people with non-O blood groups. These proteins are essential for clot formation, and higher levels are associated with a greater tendency toward thrombosis. This helps explain why type O has been linked to a lower average risk of venous thromboembolism, including deep vein thrombosis and pulmonary embolism. The trade-off is more subtle than the idea that every cut will bleed longer. Most people with type O clot normally and never experience unusual bleeding. However, population studies suggest that some studies have reported a modest association between type O and bleeding risk in specific clinical settings, but findings are not uniform. Blood type is only one part of the picture. Age, medications, smoking, obesity, pregnancy, inherited clotting disorders, surgery, and medical conditions often have greater effects on clotting risk. What appears to be a simple letter is one layer within a larger biological balance. In practice, ABO status adds context to a person's clotting profile, but it cannot determine what will happen in a medical event. The next time you see a blood type written on a medical chart, remember that the letter and the plus or minus sign describe different parts of your blood. The ABO letter reflects specific antigens on red blood cells, while the H sign usually indicates whether the HD antigen is present. These markers are not medals of survival, and they do not reveal a complete personal ancestry. Still, they belong to an ancient biological system shaped across long periods of human evolution. Over generations, populations moved through changing climates, encountered diseases, mixed with neighboring groups, and carried genetic variations into new regions. Some variations may have influenced survival in particular environments. Others spread through migration, chance, founder effects, or population bottlenecks. No blood type represents a stronger, purer, or more advanced form of humanity. Each reflects one small branch of a much larger genetic history. So that simple label on a chart is more than an administrative code. It is a practical medical tool that helps guide safe transfusions, and it is also a reminder that every human body carries traces of countless generations. Your blood type does not tell your entire story. It connects an ordinary clinical label to a much longer record of inheritance, movement, adaptation, and survival. This content is for educational purposes only and is not medical advice. If you found this video helpful, please like and subscribe to support the channel. Thanks for the support.